Now we got the light, and now this thing seems to be balking on me. Oh, it seems to be reversed. Down is...
Bottom one is forward.
Forward, okay, great. All right. So, mechanics and physical biology has recently come back into vogue, I think. It's amazing. And there was a time when I didn't think I'd see it. But if you go back in the history of developmental biology, physical biology and mechanics was one of the key things. And the old embryologists, back in this developmental mechanics, that's what this huge German word, Entwicklungsmechanics, mean, the classical school of developmental mechanics. They described the cell and tissue movements that they observed in embryos as best they could, and they didn't have a lot of tools to do that. And they tried to infer forces from these movements. They didn't have measuring things was pretty rare in those days, and so there are a number of people that had various theories that they developed about how morphogenesis occurred in development based upon the movements. And they tried to test these hypotheses about forces by microsurgically isolating pieces of embryos. They'd take it out and see if it did the same thing it did in the embryo. They'd graft it from one place to another to see if the behavior of that region was regionally autonomous. But that was foremost in their mind. And they tested hypotheses about tissue interactions at the same time. In other words, the famous experiment by Spemann, where he grafted the organizer of the embryo to another one on the ventral side and got twins, is basically the same kind of experiments. And they distinguish between material determination, differentiation of a particular tissue type, and a dynamic determination, a determination of a movement. And so inexorably linked to making the head different from the back and so on and so forth, was the idea that also movements were patterns. So they were very cognizant of these sort of things. And they didn't have computational models back in the day. They made mechanical models with rubber bands and brass rods and swelling gels and things like that. There's one very interesting one on vagination made out of brass rods connected by rubber bands and struts, brass struts between planes for bending of a cell sheet. They made a few attempts at measuring forces with magnets and bending of needles. Waddington, for example, Selman and Waddington, they looked at the neural folds rising. And they put two little dumbbells in between the neural folds and pulled on them with magnets to try to measure the force, calculate the force with which the folds closed. And they didn't do too much bad a job of it. But then time passed, and between this school and this school, there was sort of a developmental physiology period. But in the late 60s through the 80s, the dawn and the rise of molecular genetic analysis of development happened. And basically, this was focused more on a compositional mechanism, identifying the essential genes or molecules by drug interdictions, genetic manipulations, and molecular interdictions when we got better and better tools. And once they discovered restriction enzymes and were able to manipulate nucleic acids, this really hit the heyday. It hit its heyday. And the focus of all this was identifying pathways of components. And there was actually less, and still is in some quarters, less focus on how the machine actually works. And I think this is a really important thing. We started to use this word machine a few years ago, and a lot of folks are really perplexed at it. What do you mean by machine? Well, an embryo is a machine, and it's a machine that operates in order to change shape, well, speaking teleologically, to make an adult, which then in turn brings the cycle around again. But the idea of mechanism for a long time was identifying components connected by arrows and genetic pathways. And one time I was on a rant, as I'm very often am at meetings, and Geraldine Sadu, a geneticist, a nematode geneticist, she got in front of me and said, Ray, you don't understand. We have no idea what those arrows mean. Because I was taking all the arrows, I was taking all the arrows seriously, and geneticists are willing to draw these long arrows. I'll show you a very long arrow in several places between a transcription factor and a machine that actually pulls on something. And very often you'll see this. This is the thing that you have to get used to if you're a machine kind of biomechanician or physically minded person. A transcription factor is a molecular switch. And very often they'll have a line from in pathways from a transcription factor down to myosin, a mover. And obviously there's a lot of stuff between the transcription factor and myosin. Does it encode, does it turn on the gene that encodes the myosin? Does it turn on the gene that regulates the myosin? So on. So there was a very different, it's a very different world. The analysis of morphogenesis was difficult in this period because most morphogenic molecules are used right off the bat. Myosin 2, for example, in vertebrates, it comes in 3 isoforms, A, B, and C. And they're slightly specialized in their physiology, and they are used, one or the other, or both, are used right off the bat to divide the embryo into cells. So if you knock out a gene like that, you don't get anywhere. The embryo stays round. And in many genetic screens, people threw away everything that stayed round because they knew it was one of these, they used to call these housekeeping genes, because they were the things necessary in the background, and the geneticists were looking, they were screening for things that were intelligent. But the line that did something special to get development to go. What was special about development? Well, now we know that these things are all embedded together. It's very difficult to separate housekeeping genes from the developmentally important genes. And it turns out that you don't hear that term very much anymore. How many people have heard the term housekeeping gene? Most of you, yeah. Or half of you probably. But the emphasis, it's blurred. Now there was something about this era, and I'll talk about it in a bit. The thing that was popular in this era was self-assembly. Self-assembling embryos. Now, if you think about it, self-assembly applied to an embryo is a little bit of an oxymoron. What are the choices does it have? What's your alternate hypothesis? It has to be self-assembly. But developmental biologists organized whole meetings on self-assembly. And basically, it was the idea that genes would encode a selective cell adhesion by encoding molecules that imparted a selectivity, and I'll talk about this in just a second. And then when you had these balls in the embryo encoded with things of differential or selective stickiness, you then just shook the thing, essentially. It was a parallel of mass action chemistry. You just shook it. Instead of thermal agitation you had in the embryo, it would be cell movements. So cells would run around and touch one another, and if they liked one another, they'd stay together. And then they would sort themselves into positions based upon their adhesion. And I'll talk about where that idea came from and where it's gone since. This was a convenient but a rather naive connection to gene to morphogenic phenotype. But it fit. Everybody liked it because most people in this era were molecular biologists or geneticists, and there was a very simple way to get from what they were doing to something important. And that is the morphogenic phenotype. So now, in this period, about mid-80s, there were some real pioneers in bringing biophysics into development. And one of them was Anton Jacobson. I'll talk about some of his experiments later. And at a Gordon conference where he really raised a ruckus, and I sort of got tarred with the same brush because he had a big influence on me. I got into a big argument with a geneticist there. He was very famous then, less famous now. But he told me, he said, Ray, physics has absolutely nothing to do with morphogenesis. In a fit of, I said, so is this, I mean, this is almost like, you know, the thing when you go back to the Big Bang, what do they call that, the singularity or something, where all the laws of physics break down, I guess until last week when they got the Higgs boson started. Anyway, it was almost like that's what he was, I don't know what he meant, but when I challenged him on it, so this was some sort of privileged place, morphogenesis was some sort of privileged place where physics didn't apply. He retreated, said he had to go make a phone call, which in those days meant go to the payphone, you know. Anyway, so I like to bring this out because one thing that you'll experience, I don't know where you're going to go in life, what kind of things you're going to see before you get to be my age, but inevitably science has these shifts, paradigm shifts about what's important. And there was a huge revival since the mid-'80s. I call it a recent revival, but it's been going on for 20 years or so. And there were a bunch of components to it. One is, it was a confluence of a bunch of things. One is, ironically, better molecular interdictions. That would have come anyway. But basically, now we have very good genetic and molecular perturbation mechanisms, and we've got a lot of small molecule methods that where you can make acute, these combinatorial chemists can make all these little molecules that fit into pockets in proteins and block things. The cool thing is that you can flood an embryo with them and see what happens, and you can wash them out. Put them back in, wash them out. We've done that with the drug, the SB15124 or whatever the hell it is, the nodal receptor inhibitor. And it's clean as a whistle compared to like a molecular RNA encoded perturbation, dominant negative or something. And what that means is that you can target specific times in development. And there are other methods of targeting to particular regions, making transgenics where you drive a particular RNA, for example, in a particular tissue to make a tissue-specific perturbation. But time and space control is critical in morphogenesis because it gets you around this problem of dealing with a molecule that's used universally and you want to study its role in one particular stage or time. So this was a big advance. Better imaging, image processing, digital image processing. Shinya Inouye, Bob Allen, a bunch of pioneers in developing high contrast, high resolution ways of imaging embryos, marking cells with fluorescent tags, the whole GFP, development of GFP, and before that, the first dye marks that we used back in the 80s were fluorescent lipids, fluorescent dye, lipid dyes. And then at Berkeley, there was a peptide, fluorescent peptide that was used as a lineage tracer for developed by Dave Weisblatt. And then two guys out of Gunther Stent's lab, right up the road from me, between Gunther Stent's lab and John Gerhardt's lab, Bob Gimlick and Jochen Brown developed the first fluorescent dextrans that wouldn't pass from cell to cell and would mark a cell and mark all the protrusions of a cell so you could see what it did. And now with the GFPs and all of its variants, Nobel Prize was given for it, hugely important in marking both cells, and also since it's genetically encoded, you can build molecules that have this built in, of course, and you all know this. And you can make FRET sensors to look at relationships, molecular interactions, and activity reporters, tension sensing molecules, which other people will talk about. And then there was a biophysical perspective and methodology. Techniques to measure forces, much more sophisticated than previously, and also mechanical properties. And the idea of applying engineering principles in mathematical computational models actually allows us to construct and test relevant hypotheses. I put this in parentheses because to a large extent, the history of morphogenesis through a good 10, 15, 20-year period was a premature focus on testing hypotheses. Now don't go out and say that Ray Keller doesn't like testing hypotheses, because we've actually tested more than most folks. But the thing of it is, the one thing we learned the hard way, and it took the NIH and the granting agencies a good deal of time to learn it, but I bashed them over the head with it. I get vicious when I have to, fight for something. And we were told back, well, I'll show you an embryo moving here in a minute, and you'll see what the problem is. But basically, we had no description of a potential machine when I started out in this business. And we had a lot of hypotheses, but most of them were irrelevant. They were groundless. As Mimi Cole, a biomechanician at Berkeley, who got me into biomechanics to the extent that I am, I was explaining to her a classic idea about how gastrulation occurs, and testing ideas about testing this hypothesis. And she said, looked at me and she said, "Ray, have you ever tried to push toothpaste back into a tube?" And her point was the whole idea that the embryo would have had to have mechanical properties that were just likely totally impossible. And the hypothesis wasn't worth testing, simply just on basic physical grounds. And so what biomechanics and physical perspective is bringing is actually identifying between being able to see what happens and make a decent hypothesis to test is the key thing that's changed. It's really a big, and I could add to the bottom of this here, I was bashing geneticists before on their reticence to embrace, it's like genetics was the queen of biology. And then there were these folks like Anton Jacobson rushing in and saying, hey, you've got to get from the gene to a machine that moves things. How do you do that? And they sort of saw that the light would be shining on a different area of enterprise. At least some of them did. But they actually were also the savior of the whole enterprise, in the sense that some of the really great geneticists, one of the most imaginative geneticists in the field is Eric Bieshaus, of course, who won the Nobel Prize for patterning genes in Drosophila. And he's an avid thinker in this whole enterprise. So the geneticists, some key geneticists came around to this effort. So now here's the problem. This is a frog embryo, and I'm going to show you a movie of it, and I want you to get some idea of what the scale of the problem is and a little bit of the history of it. So you're going to see a movie of frog gastrulation and interrelation, and this embryo is about 1 2 millimeters across. And amphibian embryos of this type, they can have 10, 15, 20,000 cells. I've forgotten exactly how many Xenopus has. And most embryos are either spherical or flat or ellipsoid. And they, in the early period, it's one cell, and it's divided up multiple times in very rapid divisions, and there are variants on this until you've got a certain cell size. Some embryos can be down, if you look at a nematode, it's down to just, they gastrulate with a half dozen cells or 12, 15 cells or 16 cells, whatever, in various forms of nematodes. So the cell number can vary. And the other thing is that the early period can, in the case of the frog and the sea urchin, the cellular volume stays the same early on until they can swim around and feed. In other embryos, like the mouse, they make a yolk sac placenta very early and get material from the mother. Protein or amino acids and sugars from the mother. And then they make a real placenta. And so they grow extremely rapidly, sometimes from the very early period in their development. And this is something that many people miss, because they very often show the embryo, they change the scale on you in the textbook and in the papers. But they actually grow extremely rapidly. The mouse does this, and the chick embryo to a lesser extent early on, but very quickly it starts growing. And the reason is that the other category or type of growth is you'll have a big yolky cell and an embryo on one side of it. And they very quickly make a circulatory system, which then, earlier than in this organism, will ship things back from the digesting yolk to the embryo to make it bigger. And so growth is a big issue. In this animal, growth is not a big issue. It has the same cellular volume all the way through early development. But mouse, chick, many other embryos will show a dramatic growth. And so cell division, if you organize it, if you have a cube divided into two cubes, and you make a linear array of cells, they're going to get about a quarter, the whole array is going to get about a quarter longer with each cell division. But it doesn't have a dramatic effect. If they divide and grow, that's different. So cell division is different from cell division and growth. So this is a consideration. Now there's another sort of fundamental I want to make sure everybody understands, and that is that all embryos, when they first cleave, and the frog embryo, this is a huge enterprise. It has a huge storehouse of ribosomes and lipids and yolk for energy. And so when this massive embryo, and this is not a big one, they can be 2.5 millimeters in diameter in other frogs. When they first cleave, the cleavage furrow comes down, and then the walls of the cleavage furrow stop, and then it builds a new furrow. The walls of the furrow are made out of new membrane inserted as it passes through the embryo. And all this occurs every 30 minutes. With just a synthesis of DNA right back to division, with mobilizing this huge storehouse of nutrients. And so they secrete proteoglycans into that space, and then they make junctions at the top, adherence junctions. And then they organize tight junctions there. Now this business of making a cleavage furrow and then making a tight junction at the top of the cleavage furrow varies from there are a lot of different patterns. Okay, so up here is where your cleavage furrow is going to be glued together by your junctional complex. And see if I got some. I'll make some squiggles here. This part of the cleavage furrow then is inserted by insertion of membrane exocytosis of proteoglycans into this space and addition to membrane, like so. And this occurs then in the other in the plane of the board, and then like this, and so on. In the mouse, this adhesion is delayed until the 8 to 16 cell stage. They just divide inside a membrane, and then all of a sudden they become adhesive, and they form this junctional complex and tight junctions out here at the periphery, and that's called compaction. In the mouse. Human the same way. All embryos, even if they're inside a uterus and have a protected environment, make an outer epithelial layer. And an epithelium is a sheet of cells that has an apical side and a basal side and a lateral side. And the junctional complex will be around under the apex. Okay? And that's a universal. The junctions differ a bit, but basically it's all the same. This outer surface is non-adhesive in almost all cases, whereas this side is adhesive. Sticks to other things. Now in the frog, the cleavage happens to divvy up the embryo into a good number of cells, and then the cleavage furrow goes across like this and divides the outer surface from this outer layer here from the deep layer. And this is the epithelium, and this is a mesenchymal layer. Mesenchymal. And that's equivalent to doing this, see? And that happens in pretty much all embryos at some stage. Now these molecularly, mesenchymal cells and epithelial cells differ from one another. They have different intermediate filaments. They can have different adhesion molecules. They have different junctions. The mesenchymal cell is generally stuck to other cells and matrix by little point contacts. Or I shouldn't say point, that's the wrong word, spot adhesions. They can be quite large, but you get the idea. It's not circumapical, a pavement. This turns out to be experimentally very important because when you break the epithelium, anytime you're working with an embryo, when you break that epithelium, you're exposing those deep cells to the environment. And most embryos, the physiology of that the deep parts of the epithelium and the internal cells, matching it, getting them to behave right and be healthy is a dicey thing. It actually turns out to be best understood in mammals, actually. And that's because we focus on mammalian physiology a lot. Any questions about those? These are sort of just basics. Yes?
So you mentioned that the mass movements in frog castration and in avians, for example, are different, right? Mm-hmm. Can you be more specific about, like, in frog, how mass transfer?
Oh yeah. We're going to spend a lot of time on that right quickly. Yeah. There are differences and similarities. It depends on how you look at it. A lot of the same machines, to make a long story short. If you look at the molecular level, there's a huge conservation. The identity of the particular molecules varies somewhat. Like the particular ephrins used, like the particular nodals used to signal, varies. Like the mouse uses one nodal, a TGF-beta type molecule. The frog has 6. We don't, with some overlap of function. Cadherins, mice have a huge number of cadherins, frogs fewer. Wnt signaling molecules. There are certain ones that do something, Wnt11 and Wnt5a does non-canonical signaling, very often involved in non-canonical signaling in frogs. In mice, it's a different one. and sometimes they're orthologs and sometimes they're not. They may be actually a different category of receptor ligand interaction that serves the same function. So molecularly there's a lot of conservation, but little minor differences. Sometimes you can get one to work in another species within a certain bound. When you look at the morphomechanical molecules, myosins for example, there's a lot of conservation of that. But there are some differences. And what they mean biomechanically we don't really know yet. Get some ideas in some cases. When you get to the movements themselves, which we'll talk about right away, there's quite a bit of conservation. And embryos will pick and choose. I will show you that this frog uses about 4 or 5 big machines to gastrulate. And I'll just deal with next time, I'll deal with the ones that close the blastopore. I'm going to talk about categories of movements today. But you go to another amphibian. It actually uses an epithelial-mesenchymal transition, taking these cells here that are epithelial, apically constricting them, constricting their apices, deadhering and crawling in to become mesenchymal cells. They use that to generate the same force that this one generates by cell intercalation. So we're getting into a lot of sort of big picture things that I'll just tell you right now. The one thing that is very often conserved within a particular egg type, and I'll lay some words on you here, reproductive strategy. Every organism has a reproductive strategy. We have one. It's 1 or 2 eggs and a lot of care, a lot of time, a lot of growth, a lot of specialized behavior. This animal here got a different strategy. Hundreds of eggs. Sea urchins, thousands of eggs, tens of thousands of eggs. And most of them don't make it. And same is true here. Some store the energy in yolk, and then the gastrulation mechanism has to deal with that yolk. How does it deal with that yolk mechanically? It's a big mass. What are you going to do with it? Some frogs enclose it. Others don't. They say, hey, forget it. There's too much of it. I'm not going to enclose it. So they have the reproductive strategy, how much yolk there is, how fast the animal develops, whether it's a interuterine, attached to mom, very protected environment, bouncing around out there in the water. Flies are armored, both biochemically or physiologically and mechanically, in a tough shell. Some fish the same way. So all these things are ecological drivers of egg design. And when you redesign the egg, your morphogenesis, at least the early morphogenesis, has to deal with that. And so you get a huge diversity of use of basic machines. So you can name 10 basic machines. We'll get about 10 before we're done tomorrow. And if you go from egg to egg, you're going to be picking 3 of these and 1 over here, 3 out of the middle of the array, just 2, so on. But it's not conserved. Everybody used to say that gastrulation was conserved. The irony of it is that molecularly, genetically, it's fairly conserved. In terms of morphomechanical molecules, it's fairly conserved. In terms of biomechanical design, it's all over the map. And so one of the things that is more conserved than others is a system of forces. But the cellular process used to generate those forces can be different. And I'll try to demonstrate that for you. Any other questions? This is what it looks like.
To what extent are these cells still pluripotent?
At this stage, it depends on what assay you use when it starts over again. The ones on the back side are ectoderm, presumptive ectoderm. If you take those out at the beginning of gastrulation and put them into activin, a TGF-beta-like molecule, or a nodal, or inject an RNA encoding a nodal, they will become converging and extending mesodermal cells and bottle cells. You see this black line here. I'll just explain what's going on here. What you're looking at is a large mass of vegetal cells here, and a marginal zone around here which consists of endoderm and mesoderm. And it rolls inside, it involutes, turns inside, and if you put your hand up here and your elbow here and your shoulder down here, what would happen is your hand is now up here, your elbow is here, and your shoulder is down here. What happens is these bottle cells, that black line there is made by the cells, the epithelial cells, apically constricting and concentrating the pigment. And they become wedge-shaped. And they bend the epithelium. And they form a shallow groove. And so that invagination, and I'll explain that in a minute, makes a little depression. But then the bulk of the depth of the gut is made by that marginal zone rolling over your arm, rolling over this vegetal endoderm. Watch it roll. You see the cells roll in, and the whole dorsal part of the embryo, this is the dorsal side, this is the neural plate. And so, let's look at it again. Okay, see this region here turns inside, and it's converging and extending. It converges around the blastopore and it extends in this direction to drive the elongation of the embryo. And you can't see the mesoderm, this part, because it turns inside. After it turns inside, it elongates and narrows to squeeze the blastopore shut. But you can see the enormous elongation of the neural plate here. And the neural folds will rise and fuse to form the neural tube. Then you get the brain up here, spinal cord here. Now, in terms of determination, this part right here knows what it's going to do. That's the Spemann organizer. And Hilda Mangold and Hans Spemann got famous, and he won a Nobel Prize. Some say she should have, because they grafted that piece to another embryo down here. And what you see here happened on both sides. And so in contrast to the opposite side of the embryo, which is now the belly here, the ectoderm, which was liable at this stage, that region right there is pretty much convinced that it's going to do both mesoderm, I'll show you. This part right here, low, is going to make mesoderm and go inside and converge and extend to make a notochord and somites. And the part that was higher stays on the outside and elongates to make the nervous system.
So, do you mean that the initiation of all these mass movement of cells are by just a small subpopulation of cells, which are maybe expressing some markers, some receptors? I mean, the initiation of the movement.
Yes.
Is it can it be attributed just by a small population of cells?
Yes. The size of the piece that you have to graft when it starts over here, about that big. You put it down here, it will make a pretty good axis. And the classic experiment, you can do this. In fact, tomorrow I'll show you some pictures of it. I can show you after class. They grafted from a white embryo to a black one because they didn't have markers. Today, in the Xenopus course and the Cold Spring Harbor course, we use dextrans to label them, or GFPs. And you can actually look, in the classic experiment, they looked at the axis that developed from this side. And what they found was that most of this nervous system here came from the host. And only the notochord underneath here, and a little bit of the foreplate came from the graft. So that small piece of tissue talked to the other cells and said, you're not going to make ventral type mesoderm here. You're going to make dorsal notochordal mesoderm. and Semitic mesoderm and nervous system. And so they call that induction. And I told you that early in the whole history of morphogenesis, these guys are very physically minded. And they borrowed that term induction from induction coils in physics. So we have our legacy in embryology of the physical sciences in that. They didn't know that it was molecules that were passing from one tissue to another in those days. They thought there might be something like that. But they called it an induction because it was like, induction of...
Sorry. So during this process, how many-fold does the number of cells increase?
Okay, good question. The mesoderm right around that rim there, they divided once shortly before they start doing these movements, and the endoderm here continues to divide a bit, and this nervous system divides quite a bit, 3 or 4 times throughout this whole process. And
so does that mean the size of the cell decreases?
yes, it sure does. Yes. And the divisions don't line up. If you take a box and you cube and you divide it continually, that array will get longer. But that process doesn't actually contribute much length at all. They're not oriented. Now there's some report in some embryos that they are oriented. And they turn out to be controversial in the sense that they can't be repeated very well. Statistically, they're right on the edge. But there are a lot of cases in which oriented division does contribute to an elongation. And I've got various reviews I will give you. It's a little bit too much geometry and too much stuff to hammer you with right off the bat. But I will put it on a stick and we can put it on the server or whatever we've got. And I've tried to synthesize a lot of these sorts of things in various papers. Good question. Now, if this were a mouse, though, it would grow a lot. But even there, the growth and orientation doesn't appear to contribute a whole lot to the length. It's mostly cell intercalation, actually. Yes?
Yeah, that's my question. So basically, you're saying the total volume is almost conservative, and the elongation is due to cell intercalation?
Yeah, repackaging. Okay.
So how deterministic is this process? I have 2 frog embryos, and starting from the early cleavage stage, so I can for example, label all the individual cells.
Yes.
And then if I watch closely, trace the state of every cell and compile with,
In the frog, there are several thousand cells in here that will intercalate to make that axis, both in the neural and in the mesodermal tissue. It's not specified in detail. Every frog will be different. It's as if I had you guys all intercalate to make a longer, narrow array. You could wind up on the outer edge of the intercalated array at some point, one time, and be on that edge in the next round. It's not determined. The overall process is specified in terms of cell polarities and tractions and things, but the detail isn't. In the Isidian, there's only, instead of thousands of cells, there's 20 on a side in the notochord, the presumptive notochord. And this is work, there are a bunch of people, Ed Monroe and Odell. I'll give you the references. This is in the Isidian. And there are plenty of other references on the cell intercalation. They have a strict pattern of intercalation to form a 40-cell notochord. That's 2x as long. It's about 40 cells. So few cells, much more specification, it's much more organized. In thousands of cells, it's pretty loose. In the fly, there's less variance, but there still is a lot of variance in the fly in germ band extension. I'll talk some about that later.
But if there are these variations, then so in one case, the cell number 32 may end up, at the outside, and the other case it might be somewhere inside. Then these two cells, they will perform different.
Yeah.
functions.
This is a huge conundrum here, because in this case, and we'll talk about it some more later, you see, you have this giant, if I take this entire circle here, if I cut here and get that whole band around here that shrinks, it's called a giant explant. And this is the ventral end, the part at the bottom, and this is the mid-dorsal line, and right here is the presumptive notochord. And here's Semitic mesoderm. And this is notochord. Now, to put this in perspective, your backbone is organized around a rod, the notochord, and yours has disappeared. It's only it's only functional early on to cue the somites to make the vertebral column. Okay? So that's what these things make, the somites and the notochord. And this part here makes the floor plate of the nervous system. And so when you were an embryo, the distance from the back of your head to your tailbone in the ectoderm, the neural tissue, and then back up to your mouth or your floor of your brain in the mesoderm was very short and extremely wide. And it's that way in all vertebrates. And this pair of tissues, the nervous system and the notochord, somites, they turn underneath the nervous system. And then they converge and extend together. When this process begins here, this boundary is not decided, nor is this one. It's only a dashed line. And I can actually show you movies of it forming. But the process that drives this whole convergence and extension, and we'll talk more about this later. I probably should shut up about it now and then emphasize it later. Starts at a very local region here, the part that we grafted to the ventral side and got the process organized there. But out here, it's not specified. So these cells here don't know at this time that they will do this convergence and extension. They don't know yet. But what's happening is there's a nodal signal that passes. I'll draw this a little bit bigger. It passes anterior to posterior, like so, in the notochord. This is a notochord. We'll go over this again. And then in the somites, if you look at the somites, this is a posterior somite and this is an anterior one. And that nodal signal goes like that. It's very sophisticated. And this is a point I want to make about morphogenesis. I'll show this to you. Putting your players on the field is key. There are countless examples of this. The grand sweep of this thing, I'm going to show you how the ancients looked at it, and then I'm going to try to get you to the point where you can sort of get an idea of how we think it happens. But the way this works is, it's like a conductor, an orchestra. Da-da-da-dum, you guys do a behavior. Da-da-da-dum, another row or arc does. Then another, and then another. And that signal, we're talking about determination, that part, the mid-dorsal part up here, right there, it knows, now this knows, now this knows, and finally this knows. And if you take a small molecule inhibitor, SB 150, 124, that'll go in fast, My undergraduates did this experiment. You put it in early, it stops right up at the top. If you put it in really early, nothing happens. At stage 9, before this stage, the organizer doesn't know what it's going to do.
So when you say signal, that's a morphogen gradient? Or
Ooh, gradients. There's a T-shirt out that Scott Fraser likes to wear. It says there might be gradients. He's really antagonistic about gradients. There, gradients, I don't know. A wave, definitely. A progression of signaling, definitely. Whether it's let's just talk about signaling a minute in general terms. The way people think about this, it could be an extracellular gradient, an extracellular progression of a signal. It could be made here, and there could be a sink over there, and it could be degraded en route somehow. And if you do the right dynamics of that, you're going to get a gradient, and they could respond differently across the whole spectrum. Now, that's probably not what's going on with the progression of these behaviors actually being activated. But I'll tell you something else about those behaviors that go around the margin. If you look at any cell in here, I'm going to show you an experiment tomorrow that will tell you that this cell knows that this is anterior and this is posterior. And some folks think that is a gradient. That you sense a gradient and you sense a gradient. You know that this is anterior, this is posterior. This is anterior, this is posterior. And you can't intercalate. Actually, the gradient would be going the other way. And you can't intercalate unless you're faced back to front. If you turn those cells around to where they got front to back, front to front, and back to back, they will not intercalate. So they know something about that. And that's more likely to be due to a graded signal. And some people think it's a wind, but we don't know. But getting back to the idea of a progression of onset of the behaviors, you could have this signal that passes across, for example. It could be that it's made and degraded in some fashion. You could have a homeogenetic, what they call a homeogenetic propagation. You make nodal, signal to you. You make nodal, signal to you. You make nodal, signals to you, and so on. And then the signal might be degraded behind, and so it's propagated from cell to cell. Now, what's actually supposed to happen here, there are 2 papers about this signaling. One is by Branford and Yost with regard to the notochord. And that's actually dynamically the best one, though there is one in the zebrafish by Alex Shearer. And what these guys show is that, and this is known from other systems. So nodal feeds back and turns on nodal. Nodal turns on lefty. And lefty inhibits nodal. And what happens is, where the process begins, nodal is made and lefty is made. And nodal both turns on lefty, and lefty inhibits nodal. And let's see, is there another loop in the cycle here? But nodal turns on itself more than it turns on lefty. And so the way the dynamics go, it looks like nodal dominates, but is progressively less dominant over the amount of lefty until you get out to a certain distance. And then lefty takes over. Nodal signaling stops. And that sets the upper limit, the AP length. And what Branford and Yost showed is that if you stimulate nodal, if you overexpress nodal, you get more notochord faster. In other words, the stuff that the progression going up it will actually come outside the blastopore. You get too much of it. I'll explain that later. If you inhibit lefty, you get the same result. If you overexpress lefty, you get a shorter axis.
So as we see, there are different factors involved, like cell is dividing, cell is migrating, something like that. If we eliminate one factor which is responsible for cell migration or the pull, and we provide the pull from some mechanical source, so will the cell intelligent enough that it can differentiate that this is the mechanical one or not the chemical one? Like this pull is, some protein would be involved in all this, or some transcriptional factors are involved in this. And after there is a pull, then there is signals to be stopped, shut off. But if I eliminate that factor and I provide that pull with some mechanical force, will it be in a normal cell, or what will it?
We don't know. But I can tell you this, this is why you folks are at the cutting cusp edge. You should be very excited, because this is exactly where we should be today. I talked about this revival of applying physical biology to development. This is where it is. I think it's worth priming you with this idea before I actually get to the... Because you guys are coming up with all the right questions. You should be teaching me instead of me teaching you, I think. This whole business of, we call this behavior that drives this elongation, MIB. Medial lateral intercalation behavior. You don't have to worry about it now, but basically what it does is the cells become bipolar and they intercalate in arcs. But the way that those arcs progress, they go along from anterior to posterior at the edge of the notochord. This is the notochord. So they go from anterior to posterior like that along the edge. And the same out here in the somites. It originates out here laterally, and then it progresses medially. Well, what it is, well, and I'll show you measurements actually. When these arcs are forming, right off the bat, they try to pull the ends of this whole thing in. They can exert about 1.5 micronewton of force. And so this whole thing is under tension. And if you cut it anywhere in here, it will gape. It'll tear. And the progression from this lateral origin goes medially in the direction, if you look at this cell here, it's a big old cell with protrusions out on both ends, and it's attached to another one. And they're all pulling on one another, so they're under tension in this direction. So a lot of folks wonder, does that potentiate signaling? We don't know. Does it have anything to do with specification? And we don't know for sure, but it's a very attractive idea. One of my students, remember I told you in response to Roger's question, or somebody's question, on the back side here, that animal cap ectoderm, if you put that into nodal solution, or express nodal there, Activin is the one they use because it's easy to get. At stage 10.5, you won't get much mesoderm out of it, and you won't get much of this convergence and extension. And by stage 11, the show is over. In fact, a lot of folks say that the show is mostly over by the onset of gastrulation. But if you take stage 11 animal cap cells and stick them down in to this array right here, labeled, okay? We're out here and have them labeled, and look at their behavior. When that progressive wave comes through of onset of MIB, those animal cap cells, there's a delay, but they will become bipolar and behave the way they should. So the signaling in that machine there is extremely potent, far more potent than just adding nodal signaling or activin signaling as a chemical or as an express molecule. Now what's the difference? So thinking critically about it, it could be cofactors that are not expressed in the animal cap, making the tissue much less responsive. But it could be the tension. It could be the tension. But they adopt the behavior. She's basically, the student who did this is Carmen Domingo. Carmen. She's at San Francisco State University. Professor there. She did this experiment where she took animal cap cells, stirred them around, and dumped them into an array that will intercalate, and they adopt the bipolar behavior in the progression, which means that there is a signaling system here that specifies this progressively. And if you go get it from the lateral region of the embryo and stick it, or any place in the embryo, and put it back in there, the cells adopt the local order. So they can have their mind changed about when the onset is. There are a lot of interesting experiments to do there.
Great. Technically, we'd be starting discussion now. But since there's been a lot of discussion anyway, I would just suggest you continue for another half hour.
Okay. I like doing it this way.
Yeah.
Because, and I don't have to get through all this today. I can, because you've hit on a lot of things I'm going to talk about the next day. And it's good coming. This always happens in the famous embryology course at Woods Hole too, but not quite as interesting. So, okay, I'll go through some other things here. Okay, this is just basically history. Old Johannes Hofreiter, he pointed out that he was critical of Walter Vogt, the guy who mapped all these movements in another species, that this guy couldn't wrap his head around this. And it is hard to wrap your head around all these movements. And he said that, well, you can't possibly explain this as you need a superior force, perhaps. And they knew in those days about amoeboid movement. And so Vogt was imagining that all these little cells here, the cells are about as big as the dot, the laser pointer, that they couldn't possibly get organized to do this sort of stuff. And this is what it does not appear if the cells are working in the same sense that single part movements were combining to form the movements of the masses. Because the most natural and plausible explanation by amoeboid motion of single cells fails utterly. Yeah, well, you can take that view. Hofreiter was a very dynamic guy. I met him and knew him. He died some years ago. But he was very bold, and he believed in progressive reduction and reintegration of parts. And this is the key to sorting out these complex systems. You take it apart, you see what it does, you put it back together and see how much you get back under different conditions. The sum is greater than the whole is greater than the sum of its parts. It definitely is true, and there's a good reason for that. And you're going to see countless examples of why it is. But this is what he believed in. You'll get a copy of this and you can read the details. So he spawned two great ideas, and one was tissue affinity, a means of embryonic morphogenesis. And what he did was he invented a solution, his culture solution, which we call Hofreiter solution. And this was back in the 30s and 40s. And he combined that presumptive ectoderm that was on the backside of that movie with endoderm, what was right out there in that plug that got covered up. And these, fundamentally, the gastrulation takes 3 layers, ectoderm, mesoderm, and endoderm, and organizes them from being contiguous with one another on the surface of the embryo in most organisms to being 3 layers. An inner tube of endoderm, an outer shell of ectoderm, and a middle layer of mesoderm. And what happened, what happened in this experiment, they stuck to one another early on. He did this at the blast stage, and then they started to separate, and they self-isolated. They developed a negative affinity, he called it. If he did it in a little bit different array with a big shell of ectoderm here and some endoderm, and he put a little mesoderm in there, just a little mesoderm, what would happen was these two would self-isolate except for their lateral, except edgewise here. They would form an epithelium and another epithelium of endoderm. And then this would make a mesenchymal tissue in here that was space-filling. It would make matrix. And it bound together these two. They wouldn't self-isolate. So he said there was a positive affinity between both mesoderm and endoderm and ectoderm. And you could go on and make a particular array of tissues here that would mimic an embryo with ectoderm on the outside and endoderm on the inside and mesoderm between. In this animal, this rim around here is actually endodermal. It's already on the outside, and the mesoderm, most of it is deep underneath this. But the mesoderm turns inside underneath the ectoderm and applies itself closely to that inner surface, being equivalent to the ectoderm being here and the mesoderm here, except there would be a lot of specialized divisions of it. And then this tube is formed by this rolling inside. But these guys were fascinated with this possibility that there was a cell recognition. Basically, all this, they said, was driven, Hofstadter proposed, a system of affinities. They could be different. First of all, these cells and tissues would recognize one another as alike or different. This gave rise to the concept of cell recognition. And this recognition could result in either positive or negative affinities. And you had examples of a positive one between mesoderm and both other tissues. And then he discovered others, like the epidermis isolates from the nervous system in the brain. When the neural folds rise, the lips of the neural folds fuse with one another and separate from the epidermis, for example. And then later on, the eye separates from the brain, lens stays with the eye, the lens separates from the epidermis, and so on and so forth. This actually works. And I've done many of these experiments myself. So these developmental affinities are developmentally regulated. Genes determine in the course of development how they change. And that's well established now. Hofstadter followed on with work, it basically was work by Wilson on sponges, species-specific sorting in sponges. And he and Wilson basically founded this idea of selective cell and tissue affinity. And later that was transformed into what I call the school of differential adhesion. And there's one nuance to this, and that is that if you take mesenchymal cells of the three types, and these are the three colors of germ layers, blue for ectoderm, yellow for endoderm, and red for mesoderm. And if they're non-epithelial, they'll sort like this, because the ectoderm is the most cohesive, the mesoderm the next cell, and endoderm is the least cohesive. And basically the tissues act like liquids with a surface tension. And their stickiness determines this. So this tissue here is the most sticky, and the whole thing is most stable when you maximize the highest number of strong adhesions, making a sphere. Now the nuance is that if you put an epithelial tissue in here, the epithelial ectoderm, it has a non-adhesive outer side. Remember what I said here with the epithelium. And so its average cohesion is very low. Its average adhesion is very low, and nothing wants to stick to this. So it turns inside out with the mesoderm then spreading on the inner surface of the ectoderm. And you could mix up tissues, as Hofstadter did in another paper in 1955 with Towns. If you dissociate these cells but have the epidermis and epithelium mixed with mesoderm, they'll sort out and then make an outer shell of an epithelium with the mesoderm on the inside. And you can put epithelial endoderm in here and make a gut, actually. This gave rise to Mal Steinberg's idea of differential adhesion. And this is a lot to go over here, but basically he tried to make a thermodynamic model of this. And he saw these as tissues as liquids. And basically, the adhesiveness determined the order of arrays that they would make. And we can go through this in more detail after a while if you want. But if you go across here, fewer heterotypic contacts in this direction, more heterotypic in this direction. And look, he basically tried to make a work of adhesion model here that would drive the reassociation of all these things. And so if the heterotypic work of adhesion represented by these crosshatches here was greater than the average of each of the homotypic, then what you'd get is maximization of the heterotypic contacts. If it dropped below but greater than the lowest work of adhesion here, above that, then you'd get this island in the center here. If it dropped below this one, the lowest work of adhesion, then you get this self-isolation, progressively stronger depending on how far below it was. So there's a lot here about this, but the one thing we can talk about it at length, I think. But the key thing here is Hofrader talked about the concept of affinity, and that old dude was smart. He was actually an artist as well, and he had a gut feeling about things, an intuition. And he was originally afraid of this idea of just raw adhesion, and he turned out to be right. He sold out in the Townsend Hofrader paper in 1955 and called it selective adhesion. The difference is, I won't say any more about this now, the difference is that Steinberg said that it was a matter of mechanical competition, raw adhesive strength, and that one cell would be like you would be a different. You would have a choice between associating with you or you, and it would be the adhesion between the two of you would be stronger than the adhesion between you two, and so it would be a mechanical competition. Eventually, you would sort with this side, even though you would adhere it to this side, but it was sticky against sticky. Affinity allowed for something very special, and that is reach out and touch someone with special receptors, signal back in, and decide, I don't like that contact. And that's actually, this whole business does work. I hope I got this in here. And Steinberg and Takeichi did it a paper. This is the famous Takeichi of cadherence. They did it a paper much later in 94. This was in the 60s, in which they took L cells that don't express cadherins and expressed different amounts of just one cadherin. And they got this based upon just quantitative differences in adhesion. So it does work. Reed, Rouhani, and Winkelbauer, you'll have this paper. I can't go into this in too much detail now. I'll try tomorrow. Do you all know what ephrins are? Ephrins and EPH receptors. It's a bidirectional signaling system. Very complicated, and there are a whole bunch of ephrins and categories of ephrins, A ephrins and B ephrins, and their component receptors, cognate receptors. And what they do is they mediate cell-cell interactions as a physiological process. And what this paper shows is that the place where the mesoderm that turns inside and spreads on the ectoderm, that's supposed to be a differential adhesion. It's supposed to be a very strong adhesion, spreading on the inside of that adhesive ectoderm. From way back when I was a pup, I tore over the animal cap and peeled it down. And that tissue zone there just separates so easily. But that whole big mass of cells is crawling up on that surface. Why is that? The static adhesion is very low, but the behavioral spreading is tremendous. What these guys show is that it's Efrain signaling. So what happens is it's a dynamic adhesion. The mesoderm reaches out, touches the mesoderm, says, oh, I like you, I like you. I will hold and I'll pull. But then they say, but I have to detach. So it mediates cell walking. And it's not the mechanical competition at all. It's smart molecule stuff. And so here's the big dichotomy. The difference between just a raw competitive physical process and a smart physical process. And a lot of biomechanicians don't like this idea. They love to model this. And it does work as a basal sort of thing. And primevally, it probably was one of the really big organizing principles in getting like mesozoans to make germ cells inside and epithelium on the outside. Stuff like that. And you can recapitulate it. And there's another paper that I can't talk about because I'm not sure it actually is on Medline, I think, on the central Medline by these guys. And they show, it is out. They show that boundary formation in embryos can't be done just by expressing cadherins, that there's some modifier of cadherin adhesion, and it's probably Efrain's.
So cell sorting seems to be similar to like the phase separation process.
Yes, exactly.
And if the major player is the cadherin, then I guess it's going to be very difficult to have sorting into 3 different or more. Maybe 2 different types of cells may be possible. So are there any like more complex mechanisms, like different cadherin isoforms?
Yeah. Steinberg showed multiple layers, and so did but whether, there's a big question about whether or not you could do that. There are a lot of different cadherins around. So there's 2 questions. Can you do it with quantitative levels of one cadherin? Or do you need 2 different kinds with some sort of selective, total selectivity, no adhesion or strong adhesion? That's a good question. Can we argue about that over lunch or whenever? Because, it's a very interesting question. And I think a lot of possibilities for modeling it and trying to decide, I guess. The other big thing that Holtfreiter started, cell motility and behavior, also means of embryonic morphogenesis. That idea that I just presented to you was hugely popular with molecular biologists and geneticists. As I told you, all you had to do, gene, cadherin gene, cell surface, differential expression, boom. And motility, they actually showed this. If you inhibit motility, they won't sort. So instead of thermal agitation like you got in molecules interacting, you got cell motility to sample the contacts. In other words, if you can't move around and sample the contacts, you can't sort out. That was the whole idea. Very simple, gene to embryo. Holtfreiter did other experiments that everybody ignores, and that is behavioral ones. He worked on this big axolotl or other species of amphibians, actually ambystem. Maculatum punctatum, jeffersonii, a bunch of different ones. It has a different kind of gastrulation, as you'll see tomorrow. And what he noticed is if you section this thing through here, there's an invagination formed by cell shape change here. The cells become wedge-shaped, and they bend the epithelium, but he emphasized that they looked like they were crawling in. Never mind what they were crawling on, but when he isolated the cells out into culture media, the inner ends here, at the opposite end of this apical end here, would migrate across the dish. And so and then he looked at how they were organized. These ends here normally were connected to what he called the surface coat, but it was actually just part of the epithelium. So his concept was they showed directed migration and they towed everything in by virtue of connection to this epithelium. And there were a lot of concepts here. The concept of a surface coat, he was actually talking about epithelial sheet organization, those circumapical junctions. And he couldn't see those. But he got the idea of mechanically integrating cell behavior. In other words, if you've got force generated one place, it can be transmitted through the tissue to other parts. And then the concept of cell polarity and polarized motility. the concept of apical basal polarity, although he didn't know what the apicalness was. He thought it was the surface coat, syncytial surface coat. Amazing number of ideas came out of this. And he was the principal founder of this school of cell motility and behavior. And there were some other, I'm just going to take this to the embryo now. There were others that followed. Louis Wolpert, whom you're probably familiar with Gustafson, and also a number of students of J.P. Trinkus and Dave McClay and others. They worked on the echinoderm, which is clear, and they made models of how cell shape change could bend in the epithelium here and make an invagination. And also the migration of these secondary primary mesenchyme cells. And then Michael Abercrombie worked in cell culture, and this is where culture and embryos met, really. He didn't work on embryos much, but some of his students, Ruth Bellairs did, and a guy I'm going to mention in a minute. This guy really made a science out of cell behavior in culture. And what he said was, instead of just looking at this vaguely as behavior, as cells crawling around, he actually quantitated their behavior and defined some rules. Contact inhibition of movement. When cells bump into each other, they freeze when they touch one another. Contact inhibition of protrusive activity. And then contact-induced retraction. They'll pull away, and then they'll sprout a protrusion in a new direction and move off. And he also defined the concept of persistence. If you plot how far cells go before they make a turn, it varies. Some have great long runs, few turns per unit time, like that. And others go back and forth like that in some sort of quick little dance. And so this is inherent to a particular cell type. Other things like contact-induced spreading. The thing I wanted to leave you with, Michael Abercrombie, working in cell culture with his associate Joan Heisman and his tech Susan Pegram, they basically wrote the book on quantifying behavior. And then this guy, J.P. Trinkus, sort of took it into the embryo. And this is old Trink. He's a very colorful guy, had a great lust for life. And I want to advertise this book to you. It's out of print, but you can find copies. He basically looked at behavior driving the embryo, but he thought about the forces. He wasn't a biomechanician, but he knew they were important. And he studied motility and culture mostly by his students, and then applied these lessons to analysis of movements in vivo, mostly on the teleos fish. And I was in his lab then and started working on frog. He was strongly influenced by Michael Abercrombie and this analytical approach. So we got these two ideas. The idea that what cells do matters. In other words, motility is not generic. And believe it or not, there was a period in this field when if you proposed cell motility, looking at cell motility in a grant, they would say, well, we know cells crawl. And it's better studied in culture. You're never going to learn anything in the embryo. But it was considered to be generic because of this concept that only thing it did was stir the cells around. This was before a lot of cell polarity and special toys that regulate cell behavior. Let's see. Somebody keep track of the time. We're about done.
I probably want to wind up in a few minutes.
Okay. So in the coming tomorrow, basically we'll take off on analyzing a lot of bits and pieces of this, and we're going to go from small bits to big bits. And so you can look at the cytoskeleton, and it defines various sorts of things that it could be involved in, generate force for. At the next level, you can look at the whole cell, and it can show various kinds of stereotype behaviors. And beyond that, that gets organized then into big arrays. And these can be patterned along axes of the embryo, as we were just talking about before. And so you got a lot of issues here. You specify the machinery that generates force, that then makes the cell do certain things. But then you've got to get that behavior. Going from this step to this step requires a lot of signaling, because you've got to organize the cytoskeleton in peculiar ways. And then you've got to get this behavior all lined up with respect to axes. How you put your players on the field is critical. for your generating the kinds of forces that you need. And then you get multiple, you get tissue interactions. Here we've separated out two convergences in the extension, one neural and one mesodermal. But in the embryo, they occur one underneath the other. And then finally, you put the whole embryo back together. So if you work down and up in both directions, is how you figure out what drives this. Now, I think I'll start here tomorrow. What we're going to do is go through a lot of different morphogenic movements. We're going to do invaginations. We're going to do collective cell migration. Looks like we're going to have to do fewer examples than I thought. And then we're going to do cell intercalations and get into the nitty-gritty of both the biomechanics and what we know about the signaling. But you guys have actually jumped ahead a good ways. And one of the themes of this is going to be, I'm like Church Lady. Do you guys know who Church Lady was? Most folks don't know who Church Lady is. It's a Saturday Night Live. There, Dana Carvey used to have a character on there, a very prim. She had hair exactly like mine, actually, and she's very prim, and she had little pinched glasses and a little pinched nose, and she was always the person that it said, well, now, you know, so-and-so did this, and so-and-so did. Now, who can we blame this on? Satan, you know, give me an S, Satan. And there was this parodying, you know, some, sort of church lady type that was always upbraiding people for their moral lapses. Well, I'm Church Lady when it comes to morphogenesis, and I'm going to take you through a lot of special things about morphogenesis, because when you guys go out there to analyze morphogenesis, think outside the box, because the subtext to the whole business is that if you're tweaking genes and specifying things like strengths of adhesion, polarities, matrix stiffnesses, and so on and so forth, the fitness of that is solely described in terms of morphogenesis by the biomechanics of what's going on, the geometry and the biomechanics, both the material properties of the system and the geometric properties. And if it works, the embryo is going to do it. And I'm going to show you several different ways that the embryos generate the same forces. So the bottom line is, it's really a trickster. That the evolution of morphogenesis is full of tricks, and you constantly run into, out of the box, totally. You couldn't dream this stuff up. It's like a lot of things that stupid things that people do that make national TV. I mean, you couldn't write a script for it. Well, you couldn't write a script for the cute things that morphogenic mechanisms will come up with. The evolution of morphogenic mechanisms. Some of them are just, oh, come on. Who thought of that? It's like post facto design to the extreme. But it's not intelligent. It's intelligent because it works. It's selected for by the fact that it works. And so when you approach morphogenesis, the more versatile, the more ways that you have of looking at things, that's what I'm going to try to get across. Think outside the box. Think of all possibilities, because usually it's one that you haven't thought of that turns out to be the most important. Thank you.
Okay, and on that, we'll take a 15-minute break. But I want everybody